Me. Thank you for the kind introduction. I always think my mom wrote that. But anyways, thank you so much for having me. And I'm going to talk a little bit about clinical trials and what the research data is showing and some of the more um exciting developments and also somewhat controversial. And so I'll go through some of that and with a background of Alzheimer's disease. But I really want this to be kind of fun and interactive. So we'll have questions at the end. But I really want this to be useful and helpful for people because I think it's complicated and I don't think it's an easy story to tell. So we'll kind of start from the beginning and go from there. Just a little bit about me. So if people don't know, I'm at the Pickup Family Neuroscience Institute. I've been here since October. And since I've been here, it's rained a bunch, and everybody says this is what we need. But my impression of Southern California has been kind of skewed, I think, with all the weather we've had. But I'm enjoying it, especially coming from Las Vegas, a beautiful location, and I'm really enjoying being part of Hoag Hospital. If folks aren't aware, it's a great hospital system, and the Neuroscience Institute comprises a number of different specialists, and I think they're all very high quality, and I think the hospital is very high quality. So just a plug for Hoag. So we all kind of know this intuitively, but when we look at the numbers, when it comes to dementia and Alzheimer's disease, it's just a staggering disease, and the costs associated with the disease are incredible. The hard thing about Alzheimer's disease is how to quantify the challenges associated with the disease. A lot of the costs are associated not so much with the medications and the cost of care, but the burden it places on family members who are leaving work early, missing out on work, and other aspects of the cost of dementia is embedded within these metrics that are hard to get at. But when people have looked carefully, we found that the global impact of dementia is staggering. Where Alzheimer's disease is mostly going to be occurring over the next couple decades is mostly in parts of the world where medical care is not quite up to the speed of what we have in the United States. That's a controversial statement, but I think we're going to start to see a lot of dementia really affecting areas where folks are living longer, because we're seeing that Alzheimer's disease is a disease of human beings, and it seems to be very common across countries and across regions in most ways. In the United States, the desert Southwest and the Southeast are areas most heavily affected by Alzheimer's. This is where the number of cases are increasing, primarily because people are moving to the States. And so we have to prepare our healthcare system to handle the demand of the care that's going to be involved in caring for folks with dementia. We used to think of Alzheimer's as being primarily a genetic disease, but as we've learned over time, and especially over the last 20 years of our research, that Alzheimer's is less of a genetic disease than we initially had thought. And it seems to be a disease of aging. And everybody that plans to live over the age of 65 is at increased risk. It doesn't really, family can increase and protect risk, but ultimately age is the biggest risk factor. That's why it's important, I think, for everybody to think about how we're going to deal with this difficult disease. Our knowledge of Alzheimer's disease really goes back to the early 1900s, and it goes back to two individuals. One is Dr. Alzheimer, who was a German psychiatrist and neuropathologist, and had a patient who was in her late 40s. And her story was very similar to the symptoms that we see in some folks with Alzheimer's disease. And she had short-term memory loss, developed some behavioral changes, and she ended up in a psychiatric hospital. Dr. Alzheimer took a particular interest in her care because of her symptoms being somewhat unusual and the age which she developed them. And when she passed away, he was able to obtain her brain to do some neuropathological examination, and that's where he saw and described the primary proteins that we see in the brain of individuals with Alzheimer's disease, and that's amyloid and plaque. So we've known about this for over 100 years. What we've done about it is a story that's more recently emerged in the last decade or so. He was part of a really groundbreaking lab in Germany at the time, and so I've put some stars around some of the stars of the lab that worked with Dr. Alzheimer. So the golden age of neurology was around 1900s in Germany and France. And so number one is Dr. Alzheimer. Number two is Dr. Lewy, so Lewy body disease. So he described Lewy body disease. There is a couple other famous scientists in the lab who went on to make some other developments. So Ugo Cerletti was the person that invented electroconvulsive therapy. Emil Kraepelin was the first person to describe schizophrenia, and Frank Nissl was one of the pioneers of developing stains to look at the brain. So we've got a lot of what we learned going back 100 years, and a lot of frustration because we haven't really advanced beyond that. The modern age of Alzheimer's research kind of came within the last 30 years. So we have almost 80 years where very little research went on, and that's where terms like senility and hardening of the arteries. So we thought Alzheimer's disease was just part of normal aging. And in the late 70s and early 80s, it became clear that dementia was not the normal course of aging and something else was going on in the brain. And so we were able to isolate from these proteins amyloid beta, which is, we think, the cause of Alzheimer's disease. So we learned, it took us 80 years to learn that dementia and Alzheimer's disease is not a part of normal aging, and that dementia kind of comes from the emergence of these proteins that clump up in our brain, and protein meaning not protein we eat, but a collection of amino acids being protein. So our cells produce this protein called amyloid, and it builds up within the brain and it causes dysfunction within the brain. So this is what we've learned primarily only in the last 40 years. The amyloid protein itself is produced from a chromosome number 21. And what we've also learned is that individuals with Down syndrome, so trisomy 21, they have an extra 21 chromosome, overproduce amyloid. And folks with Down syndrome, because of problems with heart and with cervical spine, often did not live past the age of 40 or 50 until most recently. And so what we're learning is that most individuals with Down syndrome over the age of about 40 or 50, and some individuals with Down syndrome are living into their 60s, almost universally develop a form of Alzheimer's disease, and it's probably because of this amyloid protein is being overproduced by the extra chromosome. So these are all new developments. So amyloid is a protein that our cells produce, and it kind of leaves the cell and then it clumps within the brain. So that's where amyloid comes from. So we think that's the primary driver of Alzheimer's disease. The symptoms of Alzheimer's disease come from these amyloid plaques. The second protein that develops in the brain of individuals with Alzheimer's disease is this tau protein. So if we get enough amyloid in the brain, all of a sudden the cells start to clump together in these tangles, and these tangles are composed of tau. So amyloid leaves the cells and tau accumulates within the cells. So we have two kind of separate processes. One is amyloid production, and the second is tau tangles. So I know there's a lot of information around amyloid and tau. People are talking a lot about plaques and tangles, and that's what these are. And so it's kind of a little background information. I know it gets a little complicated when we talk about the proteins, but the two major proteins are amyloid and tau. And I think the main thing to say is amyloid plaques are plaques of cells, and tangles are cells disintegrating on top of themselves. So two different processes. So now we think about Alzheimer's disease being a disease and dementia being a stage of that disease. So Alzheimer's is a disease where the protein plaques and the tangles start forming in the brain. And they form first in that area where it's yellow right there, the short-term memory center. So Alzheimer's is a disease that starts with short-term memory problems. And as the disease progresses, other areas are affected by those two proteins. And so it starts and then it spreads. And so we see over the course of years to decades, new symptoms emerge. So in the primary course, we see short-term memory, followed by other problems with thinking and reasoning abilities, and then maybe language. And then eventually physical problems start to develop later in the disease. And that's because of the way the disease is spreading through the brain. So we think of Alzheimer's disease as now a disease, and dementia being a stage of that disease. The stage of disease is when a person can no longer kind of live independently, need some help to do the day-to-day activities. And there's many different diseases that cause dementia. So a lot of our different neurologic diseases are caused similarly. The difference is where the proteins are affecting the brain. So Parkinson's disease is very similar in the pathology. It's a protein that spreads from, in Parkinson's disease, the brainstem, so movements, and then it eventually progresses to different parts of the brain to cause other symptoms. Alzheimer's is a disease that spreads from the short-term memory center to other parts, and a disease called ALS spreads from the nerves themselves and then can spread from the top part of the brain where it's the movement part of the brain and spreads to other areas. So each of these neurologic diseases kind of acts a little different, but it has similar kind of features or characteristics, meaning it starts kind of slowly and then progresses to involve different parts of the brain. This is relevant when we talk about where the new therapies are going to be directed, because it depends on the protein in the brain. So when we talk about medications for Alzheimer's disease, I know a lot of people may be prescribed medicines like Aricept or Donepezil. There's one called memantine. And those studies we know work. What do I mean by work? When we study people who are on them versus people who are not on them, in general, people on those medicines score slightly higher on cognitive tests and in reports from families about how they're doing. But the benefit is kind of small. It's a lot smaller than we would hope. And so this is a graph of what Donepezil does for patients, and it sort of improves their score. So if there's no side effects and it's affordable, we tell patients it's probably a benefit to being on these medicines because we can get a little bit of a benefit. But when we look overall, the effect size of how beneficial, what's the magnitude of that effect, how beneficial is it for patients? It's very small and less than what we need. So I often see patients in the clinic and we start them on these medicines, and there's not a significant or notable improvement. And that seems to be the case for Alzheimer's disease. So they are beneficial, and when we study them, they seem to come out as being beneficial, but the benefits that we see are not what they should be or what they could be. So now we've moved to a stage where we want to attack the biology of the disease, because Aricept and Namenda help the neurotransmitters. They help the chemical balance within the brain, but they don't help the neurons themselves. And now we can image a lot of the biology that's going on in Alzheimer's disease. So previously, Alzheimer's disease was a clinical diagnosis. We would give people memory tests and then see how they did and then diagnose them based on that. But now, if you come to the clinic and are evaluated for Alzheimer's disease, we actually look at things like brain scans, and we may look at things like PET scans, because we can actually image the biology of the disease. We can see the neurons themselves not being as healthy. And so there's a thing called the FDG PET scan, which measures glucose uptake within the brain. And what we're seeing in these pictures is where the cells are not as healthy. And so that's because those amyloid plaques and tau tangles are affecting those cells. So now we can visualize what's going on within the brain, and that's helpful. Some folks may have had sampling of their CSF, their cerebrospinal fluid. So it's a process where we can actually look at the biology of what's going on within the brain, and we can draw that out of the spinal cord, and we can look and we can actually measure how much amyloid plaque and how many tau tangles are in the CSF fluid space. So this is another test that we can do now to actually measure how much of those plaques and tangles are in the brain. We don't often do this in practice because until recently, there wasn't a significant benefit of doing this because we didn't know how to affect this process. That's going to be changing, we hope, in the next couple of years. We have an amyloid scan now, and so we can do a PET scan measuring amyloid in the brain. This is often not done in the clinic because it's very expensive and it's not covered by insurance. So this is a PET scan, but it's not covered by insurance, but we can actually measure these amyloid plaques within the brain. So Alzheimer's has gone from a diagnosis to only being able to be made at autopsy, but to being able to be made in living people at this point. So this is an advancement of our diagnostic capabilities in the past couple of years. We have a tau PET scan. We can actually measure tau PET levels within the brain, and we can actually visualize it. So we can see these tangles. So we've come a long way when it comes to diagnostic means. The problem is these things are very expensive still. So we're not often offered because they're not covered by insurance. So what is the importance of developing therapies? Because of the ubiquitous nature of Alzheimer's disease, that it's so common in every country on the globe, we know that if we're able to stop the biology and reduce the number of people who progress to the advanced stages or who get the disease at all, we push it off by a couple of years, we can have big impacts on the number of people living with the disease. And so if we were able to stop the disease by 50%, I think this graph shows that we'd be able to reduce the number of people living with Alzheimer's disease in the United States by about 10 million. So it's a good goal to achieve. The United States government has put out a plan to reduce the impact of Alzheimer's disease. And so I think it's a shared goal to pursue. The problem is drug development is a very time-intensive and expensive process. I think on average, it takes about 10 years for a new drug to make the market, and it costs about $5 billion. So this is not a very easy process to pursue, and it takes a long time. I'm often kind of brought to the clinic, and family members will often show me results of early stage testing. And the reality of the situation is that most preclinical models, that's testing in animals, doesn't really turn out to be either safe in humans or it doesn't show a benefit in humans the way it does in animals. And that's because most of the animals that we test these drugs in, I'm talking about mice, they have a sort of a genetic form of Alzheimer's disease. And we know that most forms of Alzheimer's disease are not genetic. So this is the process we're up against when we see an animal test that's very positive for Alzheimer's disease. We know it's probably going to take at least 10 years and up to $5 billion to progress through the process. So there's a lot of different things that we can target other than amyloid and tau. And there's been a lot of work toward inflammation and how our body processes cholesterol and turning on and off certain genes in the mitochondria, but not all of those processes the body goes through are able to be affected by drugs. So this is a complicated process. This is the current state of where all the drugs are in drug development. This is a couple of years old, but it shows that about 100 drugs at one time are being studied for Alzheimer's disease, and they're in various states of development. Phase 3 is the closest to the clinic. Phase 1 is the furthest away. Phase 1 is in generally human volunteers where they're being tested to make sure that the medicine is safe. And so about 100 drugs. In comparison, I think at any one time there's about 5,000 to 6,000 cancer drugs that are being tested. So relatively small number of medicines being tested for Alzheimer's disease. So as we've seen in the past, about a 99.5% failure rate. And the question is, why have they failed? Why have all these medicines failed? Hundreds and hundreds of medicines, billions and billions of dollars. And there's been a couple of theories about why that's the case. And I think we'll walk through a couple of them. One of them is sometimes people don't think if you affect amyloid, you're going to do much. We think that maybe that's too far down the disease pathway that we can affect amyloid, that it would have a big impact on the disease. And that's kind of changed over the last couple years, but we think that affecting amyloid may have a benefit on the disease itself. But we've tried a number of different ways to affect amyloid in different ways, and so far only 2 have been successful, but a lot of medicines have been trying to affect amyloid. A lot of medicines have tried to affect tau, but tau is within the cell. And so getting to tau is a lot harder. Trying to stop that process of tau degeneration is a lot harder than amyloid. So a lot of medicines are been targeted at amyloid rather than tau. So tau is hard to get at. And we have some tau drugs, and their tau therapies have been attempted, but they have been really kind of unsuccessful. And the problem and sort of the challenge of that is tau correlates better to how our brain is working than amyloid. So tau seems to be a much more effective marker of how progressed the disease is, but we just really don't know how to affect tau like we do amyloid. And so there is a long period of time when people were saying amyloid or tau, but I think what it really comes down to is both processes are going on in the brain. So these are other areas that people have targeted and different receptors within the brain. And one of the ones I think is most exciting for a lot of people is the idea of stem cells. And stem cells for Alzheimer's disease is appealing because we think that regrowing cells would be a great way to bring back memories or to build healthy cells. The problem with stem cell therapy is, one, how do we get those stem cells into the brain? That's a very difficult process. Number 2 is amyloid and tau are pretty widespread. They're affecting pretty much all the cells in the brain. And so how do we replace those cells all over the brain without doing a lot of damage? And so a lot of the therapies that have tried stem cells have been with things like spinal cord injuries, even Parkinson's disease, where we think the disease is a little bit more limited geographically, so just a small area that's being affected. And even those therapies haven't really caught on for whatever reason. They don't seem to work very well. And so regrowing spinal cord cells, that hasn't really been effective. So the idea of being able to jump to Alzheimer's disease for stem cells is something that I think is still rather far away. And participating in stem cell studies, I think, is a little bit scary for me. And then finally, I think it's one of the things we have to think about, and this is relative to supplements. When we see a lot of commercials about supplements and the benefits of supplements, I think we have to realize and think about the fact that the brain is very selective in terms of what molecules and compounds get into the brain. We have something called the blood-brain barrier. And a lot of medicines we take and a lot of supplements we take never get into the brain because it takes a special process to get into the brain. And many drugs that have been tested for Alzheimer's disease just don't get into the brain. Either they're too big or the body breaks it down. And so there's a challenge with both supplements and medicines about whether that's getting into the brain. And so when we test, that's why we have to do testing in animals, unfortunately. I hate the idea of that. But when we tested most of the medicines, they are not able to get into the brains of animals. And so that's where they kind of stop development. So, and one of the other things is these Alzheimer's disease studies are very challenging and very long to run, and we don't have a hemoglobin A1C count for Alzheimer's disease. So, if you develop a drug for diabetes and you show that it affects hemoglobin A1C, then we could get approved by the FDA. In Alzheimer's disease and in dementia, we don't have a number like hemoglobin A1C to show that we're beneficial. So when we do studies, if anybody's participated in a clinical trial for Alzheimer's disease, there's a lot of scans, a lot of memory tests. It's very time intensive, and it's a very challenging proposition because we have to prove in multiple ways that the drug is beneficial. Whereas in a diabetes study, you may only just need to show that it's beneficial on the hemoglobin A1C, like a marker like the hemoglobin A1C. One of the other things why studies have failed in the past, I think, is that we've tested medicines a little bit too late in the disease process. And so many of the Alzheimer's disease drugs were tested in people with moderate to severe dementia. And we know by then they've already had a lot of change within the brain. And so the benefit of impacting the disease process seems to be less likely. So when it comes to clinical trials, most of the clinical trials are focusing on people at the very, very earliest stages or people at risk for the disease. And some studies have included people without using those biological markers. They've included people based on their clinical diagnosis. And the problem with that is we know that 25% of people that are diagnosed with Alzheimer's disease actually have a different form of dementia. And so we really want to be very careful about the biology of what's causing the symptoms. So that's a challenge. The last thing is that they're really hard to run and they're very long time to run. And so most studies need at least 700 participants to show a benefit. So when we see studies that involve 20 or 30 people, it's unlikely that they're going to reach their statistical power to show that they make an impact. So these studies are huge. They cost hundreds and millions of dollars and billions of dollars to run. And they take long periods of time because this is a really, in most cases in Alzheimer's disease, it's a very slowly progressive disease. And to show a benefit in a slow disease takes a long time. So these are potential solutions. On this slide, it shows maybe ways that studies can be done a little bit differently. So given that kind of background and introduction and discussion, I kind of want to move on into what is really taking up a lot of news attention, and that's the most recent 2 medicines that have been approved by the FDA for Alzheimer's disease. And we'll kind of go through what we know about them and what the studies showed. And so there's a medicine called aducanumab. And so the best way to break aducanumab down is by breaking it into 3 syllables: adu, can you, mab. And when you see a medicine that says MAB on the end, it's a monoclonal antibody. So it's a directed therapy that has an antibody right to the molecule. So there's monoclonal antibodies for cancer. There's monoclonal antibodies for arthritis. There's monoclonal antibodies for other diseases. There's monoclonal antibodies for COVID now. And so when you see MAB, it's a specialized antibody. So it's building our own immune system. So it's telling our immune system to increase its ability to detect pathogens or things that shouldn't be there. So for example, the COVID monoclonal antibody is specific against the virus itself. And so it's an antibody that binds to the virus and then it teaches our immune system about how to deal with it. So aducanumab is a monoclonal antibody against amyloid. So our body doesn't necessarily recognize amyloid as being negative. What this medicine does is it teaches our body that that's a bad process. That's a protein that we don't want in the brain. And so we infuse these antibodies into the vein. So we give this through an IV, and it binds to that amyloid protein within the brain. And it helps and then our immune system is responsible for clearing it out. And so that's what this aducanumab medicine does.
All right.
So once a month, a person goes for an
It's just interesting. I mean, I can't really walk away with anything because it's all very
subjective.
No, it's very technical.
technical. Okay, so we'll keep going. So this is an antibody against that amyloid protein in the brain. And so what this study showed was that over the course of a year, we were able to clear that amyloid protein out of the brain. And so we see these scans that over the course of 1 year, a person's brain scan goes from being positive, having lots of amyloid, to having almost none. And so that was the real big benefit of aducanumab. It's the first medicine to ever show that you could clear amyloid out of the brain. And so over time, this was a very exciting development. The problem was the drug company that developed this medicine, called Biogen, really had a difficult time running the study. And so they thought it was going to be negative. They thought it was going to be positive. They stopped the study early, and then they reevaluated the data and said it was positive. So they really messed up the conduct of the study. But based on their finding, they went to the FDA and said, look at this, we have a disease that has no clear cure, and said, look at this biological benefit you can get from clearing out amyloid. We would ask you to approve this medicine, knowing that clearing amyloid is going to be beneficial for patients. And we want to be approved based on that, because we didn't really show the clinical trial benefit, and we didn't show that it was all that helpful. And so what the FDA did was say, we will approve this medicine through what's called accelerated approval mechanism. So we're going to approve it thinking eventually it will be helpful. And so there was a lot of controversy over that, because we can't predict who it's going to help. And we don't know for sure that that medicine really helps people. We do know it clears out amyloid. The problem that followed was then the drug company decided to charge about $50,000 a year for this medicine. And so they got approval from the FDA. We know it's going to help the brain scans. We're not sure it's going to help the patients. And we're going to charge you $50,000 for the medicine. And so this is ripe for a lot of controversy. So a lot of people took this information and said, this doesn't make a lot of sense to us. And Medicare said, we're not going to cover the cost of this drug. And so that's why there's not this drug that has been approved by the FDA. It was approved in 2020, is not really readily available, and very few people were ever treated with this medicine. It also has a side effect, which is it can cause some swelling within the brain. So it's very expensive. We know it does something to help the brain have without the amyloid. We're not sure how much it helps, and it also causes a side effect. So this medicine really hasn't taken off, and it hasn't been adopted with good, I think for good intentions and good reasons why not a lot of people are treated with this medicine. But it's an FDA approved medicine. If you want to pay for it, you can get it. But the question of how effective and how helpful it is a challenge. So that was aducanumab. So right now, they're in the accelerated approval pathway. So, and we've had a lot of controversies, and I kind of described them to you here. So on Friday of last week, so just 1 week ago, another medicine very similar to aducanumab was approved by the FDA. And based on the results of a study that showed that if you give this medicine to people in the early stages of Alzheimer's disease, before they have really, really significant problems, we can see some benefit. Similarly, on the biology, we can clear those amyloid plaques. We can see some slowing of the disease progression. and they did not mess up the clinical trial. So this was a successful clinical trial. This study was recently approved on Friday. And so right now we have a second biologic agent for Alzheimer's disease approved, and that just happened. We don't know if Medicare is going to cover the costs, and we don't know what the sort of long-term side effects and benefits of this medicine are fully, because people have only been studied for this medicine for a couple years. In general, we think about a 25 to 30% slowing down of the disease progression. That's a really hard thing to quantify. What is 25 to 30% slowing of the disease? What does that look like? And right now it looks like that our test scores, so when you do a memory test, they're 25% better if you're on this medicine. So the question is, well, how much is that worth? And what is the insurance companies willing to pay? Because right now this medicine is going to cost about $25,000. So it's going to be up to Medicare to determine whether or not they're going to cover it. And then the insurance companies usually follow Medicare's lead. So with 5 million people affected by diseases like Alzheimer's and dementia, we can multiply the cost and see where the cost is going to come and who's going to cover the cost of this medicine. So there's a lot of challenges. The good news is we've taken some information that we've learned about the amyloid and tau proteins within the brain and been able to, one, be able to see that in living people now, and two, we've been able to kind of affect the biology of what's going on within the brain. And that's all done through clinical trials. So over the past 20 years, we've better understood what's going on with the disease, and now we're starting to affect the disease process in some way. So it's a big kind of development in our field, but still a lot of problems and challenges await. And certainly none of these biologic agents we think are cures. We have not really shown much improvement in people other than stabilization of the disease. So that's important to note, that none of these treatments have been able to show to reverse or significantly improve. It's more about stabilization. So that's where clinical trial work is right now. And so our goal, I think, in the next 10 years is to work toward better studies that are more dependent on each individual. And so we call this precision-based approaches, because we know that people's brains are not just Alzheimer's disease or not. They're the product of things like how much exercise we do, how much other disease processes are going on, how much have we used our brain, how much we keep it active, how much of our ability is tied into our brain, and then on an individual level, know whether we can affect the disease process. Right now, the clinical trials are based on groups of individuals who may or may not look exactly like each of us. So our hope is that we continue to move forward. So participating in clinical trials is an important part of the process. It's very time consuming. It can be very challenging, but it's ultimately very rewarding, because we want subsequent generations of people who are at risk for this common disease to have access to better treatments. And so this is an important part of the whole treatment of dementia, and hope we have some clinical trials that we're running, and it's important we match people to the appropriate clinical trial. So if anybody's interested, we can have screening visits and things of that nature. We can get folks in contact with that. But I think the idea of advancing for more effective and better treatments really is an important goal of clinical practice. So overall, I think we've made some progress. It's been slow, but we're hoping to continue to understand what causes these amyloid and tau proteins. Then they themselves are not the cause itself. There's something else going on within the brain that causes the brain to produce these amyloid and tau proteins. Can we stop that process from even starting? Right now, we're kind of set at clearing those proteins out. The question is, how do we stop that process even before that? And hopefully, and even in the future, we may be able to turn off certain genes and modify genes. There's technologies that can target specific genes. The problem is, how do we get that machinery into the brain and do that safely? So I'm hopeful that we'll be able to conquer some of these challenges. But right now, we're at a place where stopping or stabilizing the disease is probably the next step for new therapies for Alzheimer's disease. So that's kind of it for my talk. I'd be happy to go over anything that wasn't clear or people have questions.